It's a great pleasure to have Professor Itzhak Mizrahi here with us. Very brief introduction. Itzhak graduated in Tel Aviv University and then for a short postdoc in Israel in the Technion, and then since 2015 he's at the Ben-Gurion University at the Negev. He's sorry to say, but he's the international expert on the human microbiome. And if you think about microbes in cows, or how does it work, and obviously it is important there, it's mostly due to his work. And so we just spent a few days together at the international conference in Copenhagen, and I can promise you a fantastic, very, very state-of-the-art, also from technology point of view, presentation. Itzhak, it's a great pleasure that you made a brief stop in Kiel and talking about plasmids and microbial interactions. The floor is yours.
Thank you, thank you. Thank you. So, can you hear me when I'm speaking? Okay, cool, good. So, as Thomas just mentioned, usually you would hear me talking about cows and our perspective towards their impact on our globe, mainly through the microbial compartment named the rumen, the way they interact, what affects their genomes in general. Today you will not hear me talking about this at all. I will mostly talk about humans, and the reason that we got to talk about humans is actually the cows, and we'll discuss it in a bit. And the reason is mostly because in my group we're mostly interested in microbial ecosystems, specifically the ones that reside within animals' guts or vertebrates' guts. And we're asking questions about the ecology, the evolution of these microbes, what drives their genome plasticity, and how are these two components are actually interacting with each other to affect the host itself. Going back to what we usually will call in this area meta-organisms or holobionts. And we usually try to connect it to our environment. But sometimes our studies direct us to different directions, and the reason for that is a general theme that you're interested in, and we're highly interested in horizontal gene transfer and plasmids, as that's how myself and Tal actually met. So with this, I will first say thank you, Thomas, for inviting me here. It's a great pleasure to be here, and Tal for hosting me. And I will go back to the title, where I will be mostly talking about today is horizontal gene transfer, plasmids, how they affect microbial interactions, and how this overall topic is connected to the genome or the well-being of the host itself. So you probably know this slide, and if not, it's in a very simplistic manner describes what I think we should consider as a null model to think about everything and maybe to develop things from. So we have the host. It can be yourself, it could be cows, it could be fish, and the host itself has its own genome and mitochondrial DNA within it. And it is associated with what we call the microbiome, which are the overall microbial communities, including their escorting components like viruses, plasmids, and other particles that are associated with the microbiome. And there is the same microbial community that is within the environment itself. And these two are interacting. And usually when we think about these things, we don't think about the hallmark that actually makes a microbe or bacteria. So in order to be bacteria, one of the things that will make you different is the ability to transfer genes within the same generation. So we transfer our genes to our kids. Bacteria do that within the same generation, what we call horizontal gene transfer. And there are people that are giants in the field, like Bill Martin and other people, and Tal, initial work that actually made me think about these things. That study, they look at multiple different genomes, and they look at the movement of genes from one place to the other, and we understand that it has a huge impact on genome innovation. But already in 2012, when we started looking at plasmids in the environment, we realized that within a specific environment, microbes tend to transfer genes in higher frequency, and more than that, they cross way larger phylogenetic distances than we would usually expect. And if you think about this, this is great contribution to genome innovation. And more than that, if you think about the fact that these microbes can interact with other microbes in the environment, you understand that there is a level of complexity that we're usually not tapping into. And there is a person that I really appreciate and admire, which is called Thomas Bosch, that actually encourages us to embrace complexity. And with this, I really believe, because I think that there is a tendency to do reduction of ecosystems, of scientists, but we're in an era that forces us to take the broader look, to understand the top-down approach, if you would like, and then to pinpoint or develop hypotheses and pinpoint them in order to better understand the ecosystems that we're working in. And this is more or less what we're trying to do. So I hope that I convinced you that this is an important thing that is happening all the time, horizontal gene transfer. And if you think about the complexity within gut environments and the density of microbes, we are thinking about a brothel of microbes, or as we usually call it, a melting pot for horizontal gene transfer, because microbes are really promiscuous within a specific environment. They can transfer DNA particles that can recombinate into genomes, and there are some that do it in an active manner using plasmids to move them all around. So once we understand that, we can move forward and kind of shape or use this frame in order to understand the environments themselves. And what I will do today is, underneath this context, I will just introduce you to some of our recent studies taking into account this specific concept of horizontal gene transfer and its impact on microbial ecosystems and the host. So we'll start by talking about how horizontal gene transfer actually affects the host microbiome axis. I will talk about the my least favorite animal, which are human beings. And we will move forward talking about what actually drives the drivers. We know that in microbial ecosystems, plasmids, specifically in very dense ones, play a huge role. So we'll talk about what are the forces that are shaping the dispersal and recombination of plasmids within their microbial ecosystems. And we'll take this top-down approach to a bottom-up approach, trying to understand a specific plasmid and its interaction with the environment itself, an effect on microbial interactions and the ecosystem. So with this, I will just start talking about horizontal gene transfer and the way it shapes adaptivity of microbes to gut environments, and specifically to dietary polymer degradation. Now, before I start, whenever you feel like it, just stop me. If you don't understand something, ask it. I will discuss it with you. We can continue forward, because this is the point of discussions. It's a discussion. So dietary polymer or dietary fiber, we all eat them, we hear about them. Sometimes we're not fully aware to the fact that what defines dietary fiber is the fact that we cannot utilize them. Exactly like cows. Cows or any other mammal cannot utilize dietary fibers. This is part of their definition. And these dietary fibers, we highly depend on our microbes to actually ferment them and degrade them, exactly like cows. They are usually coming from the plant cell walls. They will be composed from cellulose, hemicellulose, pectin. Cellulose is considered to be the most resilient one. Then you have hemicellulose, different polymers of hemicellulose, like siloglucan or rabinoxylan, which are more elaborated, and there are only specific microbes that can actually degrade them. So essentially, in order to utilize these things, you will not digest them. You don't have any enzymes to digest them, and they will most probably be fermented in the colon, where people say that they are very healthy for you. They are healthy for you, but you cannot utilize them. And if you ask people that are working in the microbiome, human microbiome field, not the ruminant microbiome field, they will tell you that humans cannot utilize plant fibers. And they would actually be right. Most of you, 95% of you, cannot utilize plant fiber. And the reason for that is the fact that you don't have the right microbes for actually being able to do that. And recently, we actually discovered 3 specific species that we call cellulitic species, because they can actually utilize cellulose, and we find them within gut environments of human beings. And when we find them, we actually realize why humans cannot degrade plant polymers, specifically cellulose or hemicellulose. So this is just an illustration that summarizes a lot of the findings. Again, if you don't understand something, just ask me. So on the y-axis, you see the prevalence of these microbes across human populations and also apes and non-human primates. Essentially, prevalence means that in how many individuals within a specific environment we will find these microbes. The amount of the green things over here, these are the microbes, is their abundance, okay? So this summarizes what we found. We found that in industrialized humans, you have very low abundance of these microbes that can degrade cellulose and low prevalence. Essentially, the number is 4.5% of the industrialized human population across hundreds of human beings can have these microbes, and these microbes will be able to degrade cellulose. The reason that we know that, because here in blue you see machineries that we call cellulosomes that enable microbes. These are amazing machineries that were invented by evolution that enable microbes to adhere to the plant particles and degrade them very, very fast. We find such microbes in the cow's gut. But if we look at other human populations that are living now on planet Earth, like hunter-gatherers, we will find higher abundance of this species that we found, and also higher prevalence. About 20-30% of the people that executing the hunter-gatherer lifestyle would have these microbes with higher abundance. And if we go to samples that were taken from people 2,000-3,000 years ago, they weren't taken there, they just found species that they could extract DNA from, we find about 40% of the samples to actually have these microbes in higher abundance. And if we go back to our closest relatives, apes, we find these microbes with 30-40%. In a second, we'll talk about the different lineages, and with very high abundance. Essentially, this only circumstantially suggests that westernized lifestyle somehow negatively affects these specific microbes. Now, up until now, are there any questions? Because this is I'm showing you pictures, and you need to rely on what I'm saying to you. And in this context, I will try not to lie to you. So this is what we have, because it's recorded. I cannot then revert. So let's move forward and talk about the specific species or strains that we found. So I named them, and now I feel very, very sorry that I named them as such, because I can hardly pronounce it. So we have Ruminicoccus permisiens, and the reason that we named it like this is because we found it mostly associated with non-human primates, not apes, okay? The other one is Ruminicoccus hominiciens. The science is, in a second I'll explain why, and this one we find mostly in industrialized humans. Here we have less than 100 people, so most probably there are some lucky ones here, but with very, very low probability. Okay? We find it in rural societies in Eastern Africa, where they still eat sorghum, like very high concentrations of dietary fiber. And we find it within hunter-gatherers, which are the Hasda, about 400 samples, very high prevalence. And we also find it in apes. Okay? Now, these two we can find also in some of you. They are kind of cross-reacting, but none of them is found within cows. They are only found in humans. They are mostly associated. We see their higher abundance in this group or in this group, but are not found in cows. And the reason that I'm mentioning that is because the reason that we found them is this microbe, Ruminicoccus flavificens. We have these huge databases in the lab of all the mics where we want to understand where microbes are coming from or genes are coming from. So we started finding genes from this microbe inside human guts. And at the beginning it looked like contamination. And in some point Sarah, which is a staff scientist in the lab, and kind of, was created with the matrix. She's been studying these things for ever, the enzymes themselves. So she is working at the lab that invented the cellulosomes, the lab of Professor Ed Bayer and Rafi Lamed. She told me the fact that we see these genes inside these genomes means that this microbe is there. It's not a mistake. And we started looking for that, and then we identified them in humans. And as a tribute to this microbe, we named them science. Okay, so just as an homage for cows, you should understand that we didn't started studying the humans. We started with the cows, and it just led us there. So This is how it began. Now, of course, you're asking yourself, why do we have them and when did we get them? Yes, so this is what we started asking. And in order to ask such a question, what you need to do is kind of an evolutionary analysis, trying to track back the interaction of these microbes with their hosts and the trajectory that led them into our guts. So to do this, we usually take all the proteins that exist inside the microbe genome and we create a phylogenetic tree and try to see to what other microbes it actually connects to, okay? And we created such phylogenetic trees for each one of the genes that existed in these microbes. And also we created what we call a concatenated tree. So this told us two evolutionary stories. I'm presenting to you the one that is the more, let's say, probable one. So this is a tree that most of our gene trees inside the genomes of these microbes we actually got. I will tell you what is the other scenario in a second. So what can we learn from this tree? In pink, you see the genomes coming from the microbes that are mostly associated with the non-human primates. In green, you see either the mugs or genomes coming from the Ruminococcus flavificens that we talked about. And in blue, you find the genomes that we found only in humans, okay? So the first thing that we can see over here, and this is kind of, most of the trees told this story, is that the lineages in humans are diverging from the ones that are coming from cows, essentially suggesting that these microbes are descendants of what we call the Ruminococcus flavificens that we found mostly in cows, okay? So this is one thing. The other thing that we found is that some of them, one of them, let's say, Ruminococcus ruminescens, we found a lot of its genomes, is it looks like it's still inside the clade of the cows, yeah, and not clustered with its other counterparts. And it's somewhere in between. It's most probably, and you will see the other data that supports it, a recent transfer. So this was kind of invading the human gut later on after this species invaded the human gut. This is a riddle for us, but we see it. So this one is found mostly in humans, okay? And it's more close to the primate guts, meaning that it might be another transfer that was introduced into the human or invasion to the human when it's still trying to adapt to the human gut. We'll look at their genome composition in a second. To you I'm telling, and if you read the paper, you will see that there is another story told by the genomes of these microbes that some of the proteins, not all of them, show co-speciation with their hosts. What does it mean? Whenever we see a split between the humans, the ruminants, and monkeys, we see also corresponding split inside the microbes. But this has the smallest proportion in their genome, so we cannot say that this is necessarily the scenario. The more probable one is this one, that they are descended of microbes coming from cows. And of course, the romantic ones among us can say to ourselves that most probably during domestication, we somehow captured these microbes and they are still being captured nowadays. With regard to the acquisition, we think about cows, and this tells us that they somehow got adapted at the protein sequence, meaning these trees work at the protein sequence. That's how we create them. But when we think about their host association, I just told you that they are found only in humans or monkeys or non-human primates, but they are not found in ruminants. And only changes, again, it's just an intuition, only changes that the protein sequence cannot explain such adaptation or exclusive-ness of these microbes within a specific gut ecosystem. So we wanted to move forward and ask how they are adapted to the human gut, or what type of mechanisms allow them to adapt to the human gut. And in order to do that, the first hypothesis that we have after the genome sequence is what is the composition within their genomes that allows them to actually reside with a specific gut and not the other. So the first thing to do, from my point of view, is usually using an unsupervised method, a way that you do not bias the data in any way. And the most prevalent one is what you call PCA, which is essentially asking the the data to present itself, to show where most of the variance lies, okay? And then we can do all types of clustering. So in order to ask such a question, what we actually did is we took each one of the genomes and we looked at presence, absence of specific genes across the genomes with the question of how similar are they to each other. And what I'm going to show you, it's just a plot of similarity. It's not a similarity, it's how the variance is explained on the PCA plot. And this is what we got. We got the microbes that are usually residing within cows as a cluster. Okay, so these are the Ruminicoccus flavofiscens. The microbes that are mostly residing within humans as a cluster, and the microbes that are coming or residing within non-human primates as a cluster. And as you can see, remember that the two recent transfers, this is one of them, which is kind of somewhere in between the cows and the human associated microbes, somehow still adapting, and the other one is residing within the monkeys cluster. But what does it teach us? It teaches us that at the level of genome composition, presence, absence of genes, although they are different from each other, their composition, the genome composition, is quite similar of all of these microbes. And this is true when we're looking at the overall genomes. This is true when we're looking at specific genes that are responsible for fiber degradation. And this is true also when we're looking at the overall transcriptome, which is representative of them all. And this is just a clustering. Of course, they cluster in a very significant manner. So once we see such a thing, we ask ourselves how this adaptation actually occurred. How did we get from assuming that they're descendant of the cow's microbes, how did we get these two different clusters? And you can think about two very simplistic scenarios. One would be losing genes and then from the ancestor, so you either end up here, or they're actually acquiring genes from the environment, or anything in between. So we can actually differentiate by just looking at what they got and where did they get it from. Is this clear? Good. So in order to ask this, we cannot only look at what is acquired by horizontal gene transfer, we need to ask how deeply a gene is rooted within this specific phylogeny or within this specific lineage. We're good? So for this, we have an amazing collaborator called Bill Martin that kind of do the analysis and also invents the methods. And Bill created this tool, method, or whatever. It's an amazing one, which is called verticality analysis. And this analysis, what they do is asking how deep a gene is rooted within a specific lineage. So you don't only get information on whether it's acquired or not acquired by horizontal gene transfer, and this Tal can tell you way better than I do that usually you can do that when you're looking at the tips of the tree. It's very hard to look backwards. Am I right, Tal? So Bill actually, no, this is, everybody should know their limitations. So what happens in this analysis, you go deeper into the tree and you ask how deep and how fundamental this gene is within a specific lineage. So high verticality values would be deeply rooted, meaning not horizontal gene transfer. Low verticality values, it means that more probably this gene is being acquired by horizontal gene transfer. Essentially, this analysis can differentiate for us between these two scenarios that we just talked about, whether they got it from the outside or they lost genes. If they lost genes, most of them will be with very high verticality values. So these are the results. So we see that core proteins, which means proteins that are shared across all of these lineages, have high verticality values, okay? Host-specific proteins, meaning proteins that are shared only within a specific cluster, have low verticality values. What does it mean? It means that most of the proteins that makes this species host-specific, that makes them cluster close to each other, are most probably coming by horizontal gene transfer. Bill told us that this is very recent transfers, so they are capturing it from the environment all the time. So essentially, this adaptivity, going back to the framing of this talk, is occurring due horizontal gene transfer. And then you ask yourself, what did they acquire from the environment and how it made them adapt? I'm going to show you only the genes that are related to the ability to degrade dietary fiber, because this is what we're talking about. So we did a parallel analysis in order not to be biased by this one. And we find that the genes that were acquired from the environment itself are genes that are highly related to the lifestyle of these microbes. For example, you remember in pink we see the non-human primates. We see that they have exclusively this gene called GH19, glycolysyl hydrolase 19. This is responsible for chitin degradation, most probably because this, the hosts of this specific microbes utilize or eat bugs which have cytoskeleton composed of chitin. Another wonderful example, for us it was very surprising, is this gene, GH98. You see the GH98 with the dock, it means that it gets into this very elaborated machineries, and without, it's just being excreted to the environment to degrade planetary fiber. This specific gene is exclusively found within the human strains. And amazingly, it is responsible for the utilization and degradation of hemicellulose that is coming from monocots, like maize and rice that people has domesticated and has been eating for years, and it's only found here. Of course, we verified that they are actually transcribed in the environment themselves, and we also extracted the protein itself, and we did a lot of biochemical assays to see that it actually does it. So you find a specific gene that once these microbes capture it, its host can actually utilize dietary fibers that are coming from domesticated plants that they just domesticated. So with this, maybe we can summarize this portion. I hope that we understand that horizontal gene transfer can have a huge transformative effect on the phenotype of the host itself just by microbes acquiring genes from counterparts and being able to survive. This will mean, for a lot of you, less constipation when you eat planetary fibers, if you want to take it to the fine details. Or there are other people that suffer from any other consequences. And we know that the fermentation of dietary fiber at the colon can, for example, lower the probability of inflammation, because you have a lot of butyrate as a function of these specific types of fermentation. So you can think about many, many ways that you can actually introduce these microbes, and of course we are working towards these directions. Up until now, do we have questions? Good, so we'll continue with this space. So of course we talked about the importance of horizontal gene transfer and its effect on the host itself. But once you see this huge effect, and we see it all over the place also with cows, you want to ask what drives horizontal gene transfer and who actually accommodates that. And we know that from a lot of perspectives, there are many trajectories for horizontal gene transfer, but we are highly interested in these components called plasmids that probably you heard a lot from Tal about, and today I learned that other people are working on this topic. And plasmids, the reason that we're highly interested in them is because in this dense gut environments, they are playing a crucial role. And the reason for that is because they're requiring adherence and attachment between microbes. And what they can do, plasmids are selfish. Most of the time when we look at them, they don't carry any other gene except of their own maintenance, what we call maintenance genes, in a not very creative manner. So plasmids can hop in and outside of the genomes, and sometimes they can carry part of the host genome with them and transfer it to a counterpart. Once they did that, essentially you got a transfer of a gene from one microbe to the other, even crossing phylogenies. Sometimes this gene is getting back to the chromosome, as you just saw. And nowadays, we realize that there are tremendous amount of data of plasmids that we can actually extract, but we don't fully understand what drives this processes of horizontal gene transfer and the movement of plasmids from one place to the other. And with this in mind, Alva Zorea, which is an amazing student at the lab, wanted to ask what drives plasmid dispersal. So in ecology, dispersal is extremely important. We're living under this everything is everywhere for a while now, but we fully understand that there is differences in frequencies of different organisms in nature and different entities. And once you understand what drives dispersal, you understand that what are the forces that can actually govern these processes. For example, if you move around and you see a person, whenever you move, you see this person next to you, you can assume a lot of things. It's either, in my case, my wife. or somebody that stalks you, but the process is not random. It's not that whenever you move, you just see a different person. So there is some of connection, because there is no connection to the abundance of this person on planet Earth, okay? Or whenever you see a human being, you see flies around it, probably there is a causal connection. Or whenever you see humans, you can find mosquitoes, so these two things are interacting. This is a specific example for selection. But there are other forces that govern nature, which are not selection. They are not deterministic. They are more probabilistic. And you can actually differentiate between these two by thinking and looking at the ecosystems themselves. So in order to answer this question, what drives plasmid dispersal and recombination, what Alva did is the following. She took samples from 3,600 different samples around the globe. And she used an algorithm that we developed together with Ron Shamir. Its previous name was Recycler. Now it's way more elaborated. She assembled a lot of plasmids, and she went through a very stringent screening process to reduce them by clustering them, and also moving through a lot of different databases to make sure that they are actually plasmids. And we left out with about 11,000 different non-redundant plasmids. And these plasmids are coming from people all over the globe, as I just said, from different disease states. Healthy is also a disease state. And then we can ask a lot of questions on what drives their dispersal across the globe. And the way you do that is you need some type of a reference point. You need to ask yourself, what do I compare my data to? Okay? Or what is the null model? And in our case, the null model was the most obvious one to use, which is essentially that there are no forces that drive the dispersal of plasmids throughout this planet, okay? Essentially assuming neutrality. So to those of you that are not familiar with the neutral theory of biodiversity, it essentially assumes that the abundance of a specific entity, in our case it would be a plasmid, will govern their prevalence across populations. What did I just say? I said, in very simple words, that this pink plasmid that you see that is very highly abundant within the gamma diversity pool, in the actual reservoir, would be more probable to be found in more individuals, okay? And the green plasmid would be less probable to be found in multiple different individuals, because there is no selection. Their probability is exactly throwing a cube. So the probability of getting 1 on a cube is equal to the probability of getting 2 or 3 or 4 or 6, because their prevalence is, their abundance is exactly the same on the cube itself. So this is what we would expect in the case of neutral selection. Sorry, neutral selection, there is no selection. It wouldn't be the case if we had a selection on this green plasmid. We would find it in all of the individuals despite its abundance. Back to the example that I gave with my wife or my dear friends or people that are stalking me, okay? Their abundance is not very high, but whenever I move, they move with me, okay? So selection and neutrality we all understand. So now what we need to create is a very basic null model that assumes that the prevalence of plasmids across human population will be correlated to their abundance. And this is what you see over here. We, of course, looked for the plasmids that are being selected, but we were amazed by the results. Let me first describe this graph, and then we will all be amazed by these results. So first, on the x-axis, you see just the abundance of the plasmid at the gamma diversity pool, the pool that we described before, okay? And on the y-axis, you see their prevalence across human population. 1, it means 100%, okay? This is the null model that assumes neutrality in orange, meaning that whenever you have an increase in abundance, you have an increase in prevalence, okay? And whatever sits on this graph or close to it, or the fit between them, means that plasmids are dispersing in a neutral manner. So us, you know, when you think about plasmids, what do you think about? Selection, no? Antibiotic resistance. So we were quite amazed by the fact that they are distributed in a neutral manner. Each one of these dots, blue dots, is essentially one plasmid. So the first thing to understand here is that you can have plasmids in about 90% of the population, okay? And they are not being selected for, most probably are not selected for, because it's kind of correlated with their abundance. And vice versa for low abundant plasmids. It's not that they're being selected out. But in some cases, we find plasmids that their abundance does not explain their prevalence. So they would be in lower abundance, but with a higher prevalence in the population, this would suggest that these specific plasmids are being selected for. Now we're stuck in a catch-22. And why? Because plasmid, I just showed you this nice cartoon, I think it's a nice cartoon, that shows plasmids inside microbial cells. So whenever the cells are moving, the plasmids are moving. So in this case, we are confined to the dispersal of the host, of the microbes themselves. So if the microbes are distributed in a neutral manner, also they're plasmids. So in order to ameliorate this problem and then attack it from a different angle or triangulate it, we started looking only at the segments of plasmid. What does it mean? Now we're not looking at the overall plasmid. We're doing the same analysis, but we're cutting the plasmids. We're ignoring the circularity, and we're even putting them out of the analysis and looking at the segments as individual entities. And we're looking at each one of the disease states, because another thing that we wanted to verify and understand is whether this signal that I just showed you is being pulled by one data set versus the other. So to see whether the healthy are neutral and the other ones are not, and vice versa. So what we did is looking at the segment in each one of the disease states, as we just described. And as you can see, in all of the disease states, we have very nice fit to neutrality. So it doesn't matter in which system, or at least with regard to this data set, from which system you're coming from. In most cases, segments, the recombination between the plasmid is driven by neutrality. And in each one of the systems, we also find specific plasmids that deviate from neutrality, as you can see. And the interesting part is that these specific segments carry ecosystem very relevant genes in most cases. Just as an example, you see this orange part in the irritable bowel disease, the glucose-related diseases, we call them, but it's diabetic. Both of them are considered inflammatory diseases. And the healthy, you see this, they have very high proportion of this brown thing. It's essentially beta-lactam resistance. This is ampicillin resistant, which is highly found on these specific plasmids. It's not found on the ones that are behaving neutrally. And this is not very surprising, because we are being fed with antibiotics from the moment that we are born. And as you can see, maybe some of you already saw that the R-square for the healthy individuals is higher than, for example, the IBD. So this suggests that in the IBD there is a deviation from neutrality in general for the overall ecosystem. And it means that they might be experiencing harsher selection pressure. So let's say that you put antibiotics on the system, there would be harsher selection pressure. So in order to examine this more, which plasmids are being selected, and whether there is harsher selection pressure in these systems, we wanted to move forward and maybe triangulate the same exact thing from a different angle. And in order to do this, we essentially created plasmid sharing network. What does it mean? It means that if we have individual A and B, and they share a segment between them, we will draw an edge between them. So we have nodes, which are the individuals, and we have edges that are essentially the connection of the segments themselves. Just put this to see that I'm not late with time. So we can now create a network of plasmid sharing between different individuals across the globe. And in order to know whether this edge is happening by random or in a neutral manner versus the one that are being selected, we create a null model in which we randomize the edges but keep the structure of the network. And we can put a specific p-value on each one of these edges. In other words, simplistic words, I'm going to show you a network of only the segments that are being selected for or are deviating from randomness on the overall globe. We're good? Please stop me if you do not understand something. So, please.
So just a small concern.
What is your name?
I'm Samir.
We've met.
Wow. We have met, exactly.
Yeah. Congrats, by the way.
Yeah, exactly. An amazing method. Just a small concern. Did you make sure that these segments are specific to one plasmid and not shared with the chromosome or with different plasmids?
So the whole idea is, first of all, they might be shared by different plasmids. The idea here, the thinking, is to take out the plasmid components from this game and to ask the plasmid as a whole, as an entity, to ask about recombination between the different entities. So in this analysis, once you have a whole plasmid, it's not part of the analysis. We are taking it out. Only segments that are, if I remember correctly, that are bigger than 1 kb are getting in. Of course, we tried it in different thresholds. So there is a threshold that you see that. And then you're essentially getting a recombination network between plasmids. Or in other words, what we're usually looking for, how they recombinate with each other. Some of these genes could reside on the chromosomes, but the chromosomes are not part of this analysis. You're looking only at plasmids. Throughout this analysis, these are only the entities themselves. And this analysis is what they actually shuffle between each other. Why? Because already in 2012, we realized that plasmids kind of distill the most important functions within a specific ecosystem. So we wanted to see what they actually recombinate, because these are the drivers for horizontal gene transfer. Good?
Sounds very nice.
Okay, so now I remind you what you're going to see. It's edges between individuals that drive only the combinating parts between the plasmids and only the significant one. So this is what you get. Each one of these things you see here, it's an individual. A lot of nodes. They share, these edges are essentially segments that they share between each other. And the colors are according to the disease state. Here we took out the glucose-related diseases because we didn't have enough power there, so they just interfered with the analysis. And the edges that you see, as I said, are only the significant ones. So it's very clear that the green ones do not cross continents. You see that? These are individuals that are living in different continents: Europe, Asia, North America, and Africa. Okay? So somehow, healthy individuals are confined by geography as a function of the segments that they share via plasmids. This is an observation. You can think on why. Why, for example, could be lifestyle. So it's the same lifestyle, selection of the same genes. Could be diet, specific diets, and more probably the selection force does not allow transfer across continents, okay, or a combination or the same functions to be selected in different continents. But this is not the case, for example, with IBD. You see that in IBD, unlike healthy individuals, you breach this confinement by geography. So here we can assume, and of course this is kind of an observational study when we do big data, we can assume that the selection force, as we've seen when we looked at the neutral model where we had more deviation from neutrality, the selection force in this situation is harsher, so it selects more, let's say, relevant genes to the environment, or we can find on plasmids more relevant genes. And when we look at the overall gene composition or functional composition on the plasmids, we find highly relevant ecosystem genes. Just as an illustration, we find, for example, this biosynthetic pathway of aerobactin synthase on a lot of the plasmids that are being transferred all over the place. And this system is aimed at scavenging iron by microbes that are usually pathogenic, and specifically in IBD, it was found in multiple different studies. And it's one of the markers for IBD. When they see these genes there, they know that there is an inflammation, and it's being used by the microbes, usually pathogenic microbes. They need iron from the ecosystem, so they would have siderophores that will allow them to capture them. And many, many other genes like this. I must say that a lot of the genes, or most of the genes on plasmids, are actually not annotated. So essentially, just to summarize this, we understand, like, unlike we usually think, plasmids are disperse and recombine in a neutral manner. And when selection kicks in, then you can find on plasmid things that you would see later on the genomes, the most relevant genes that are important for this ecosystem. So if this ecosystem needs, I don't know, fiber degradation genes, you will find them on the plasmids. We've seen that in the rumen. If the system needs scavenging of iron, you will find that. If you're being selected all the time for antibiotic resistance, the first thing that you will see that will reflect the needs of the ecosystem would be found on plasmid, and you will find antibiotic resistant genes. So up until now, what we actually did is, not with the first one, but the plasmid one now, we looked from a bird's eye view, and we tried to understand how these ecosystems actually behave and what are the ruling forces and what are the ecological driving forces for these ecosystems. But I was asked today, how sure are you that this plasmid is actually a plasmid? And my answer was, it depends what is trust. Okay, trust could be going to a bar. I think this is what going to a bar with a person. So you need this person not to rob you or, I don't know, murder you. But when you get married, you need a bit more trust between the partners. So it depends on how much you invested or how big is your question with relation to a specific case. So in order to describe how ecosystems behave, it's good to look from a broader view to understand the driving forces. But then in order to kind of follow an hypothesis, you need to be really sure, and that's what we're going to do. We're going to drill down now and try to understand an importance of a specific plasmid that is selected within an ecosystem and to understand its impact on its microbial ecosystem. And this was done again by Sarah Morais, that I mentioned before, a staff scientist at the lab, an amazing person and a scientist. So Sarah said, okay, this whole thing bullshit with this red things, course and continents, this is big data. I like, remember Sarah with the enzymes, I like to look at something, to touch it, to experiment on this. So what Sarah did, I told her, okay, choose a plasmid and work on it. So she has very sharp eyes. She looked for plasmids that are crossing continents and are found on multiple different plasmids, or specific genes that are found on multiple different plasmids and on found in a lot of individuals and selected for, deviating from neutrality. So this was found at about 700 individuals and 14% of the cows, which is amazing. First, because it relates to cows and it might have an effect, and also cows are very easy to work with and it's an amazing ecosystem. Next time that I will be talking here, I will be talking about cows. We have some amazing studies there, like findings. So one of these plasmids that was very highly prevalent in cows, the same plasmid with this specific gene, we annotated it into the Enterococcaceae family, and we wanted to move forward and to ask questions about this specific plasmid. And the reason that we chose this specific plasmid is this gene that we thought, wow, we have something very important here, because this gene is a part of an operon that converts glycerol to propionate. Propionate is a very important compound for cows. Actually, what you're drinking, the milk that some of you are drinking, is coming due to this compound. It's responsible for gluconeogenesis. Genesis is the precursor, becoming from this thing to sugar, then to milk. And you guys can actually also use it exactly for the same reason, energy metabolism. So we said, okay, we find a plasmid that affects the energy balance of the ecosystem. Nice, no? This was the hypothesis. But unfortunately, in every plasmid that we looked at, we only found this gene, the 1,3-propandiol dehydrogenase, without the other parts of the operon. And this took over our overall hypothesis to the garbage can. And we were very sad, but when we actually thought about this and read a bit of Wikipedia, this is highly recommended for everybody, we realized that this compound, 3-hydroxypropionaldehyde, is an actual toxic compound to microbes. It's antimicrobial. And we raised the hypothesis that this gene, which is only part of the operon, might be detoxifying this compound. And as you can see, this compound is also called reoterin, and to me it rang a bell because I know a microbe previously called Lactobacillus reoteri, now Limnosibacillus reoteri. It's a probiotic microbe found in most of you right now, and I have a good friend that works on it. And then I realized that other microbes produce this compound reoterin, also Eubacterium hallei, so it's highly produced by a lot of microbes. This guy is not a farmer. He's called Professor Jens Walter. He's been working with this microbe for a while now. And I told Jens, can you please send me this compound? Because you cannot buy it. I wanted to understand whether it's relevant to the environment. We wanted to create a targeted metabolomic approach that will help us to quantify it in the environment and understand what are the concentrations. So he sent it to us. We milked the microbes for cows for this compound, and we calibrated a targeted metabolomic approach to actually measure its concentration in the environment itself. And when we did that, we found it at 50% of the cows that we could actually look at, so it's ecologically relevant. And more than that, it gave us the exact concentration, the physiological concentrations inside the ecosystem. Why is it so important? Because I told you it's an antimicrobial compound. We could use it now to isolate the microbes that have the plasmid, going back to how much you trust before you move forward. So Sarah, that's actually what Sarah did. She used the media, she used the rotavirine inside, and she started isolating microbes that have this plasmid, and she indeed found a microbe called Enterococcus faecalis with the plasmid inside, which is nice because we predicted that this plasmid is inside the Enterococcaceae family. And then we, of course, do not trust plasmids from databases. Also, we did here PCR, but we also developed a method that enables us to see plasmids within the host cells. So this is plasmid where it's distributed across the whole cells. This is high-resolution microscopy, and this is compared, for example, to a chromosomal gene where it's very, very focal. So we can actually detect it. We were sure that this is the plasmid, and it resides within the cells, and we could move forward and start asking ecological and physiological questions about this plasmid. Now we trust it, because it's there, we can see it, we can touch it. Now, what we did is we created a system, a genetic system, composed from the isolate that we took from the environment itself, and isogenic strain that we ordered from the DSMZ without the plasmid, and also the same isogenic strain where we clone this plasmid into. It's not easy to work with enterococci. This tributes are going to Sarah. Somehow she managed to clone it inside with a specific system. Then we can compare the resistant to rotarine to see if this plasmid confers resistant to rotarines. You remember this was the hypothesis based on the Wikipedia thing. So what you see over here, these are increasing concentrations of rotarine because we didn't know how much it will inhibit. And this is the plasmid that we cloned into the isogenic strain. And this is the plasmid inside the strain that we isolated. You see that they respond more or less the same to the concentrations of rotarine, which essentially tell us that the plasmid itself is responsible for the resistance. Otherwise, this one would not be resistant. Okay? Then we looked at each other. We did a tremendous amount of work just because of trust. And we said to ourselves, what? We just have a toxin-producing strain, and the one that have defense system, and this is it. For this, we did all this work. And Sarah said, yes. And I said, okay, it's a problem. It's not a problem, but it's a bit painful, because we were hoping we started with this energy balance, etc. But then Sarah she sees all the time, she has, I think now, 5 kids. She just gave birth, so I counted them from the beginning till the end, and she said, okay, I just saw this thing that you probably all saw, Nemo, and there you have sea anemone and a fish. The sea anemone produces toxin, and the fish is just there, okay? But this whole thing is a cover. It's a system that allows these two organisms to interact with each other without any other to interfere with this. So they cross-feed each other. Maybe this is the case here. I'm telling you this like this, very simplistic, but it was very painful, this process. So we said, okay, how will we verify this? We just need to grow them together and see if they grow better together like these two, okay? If you put this fish outside, it's orange, somebody will eat it, okay? So when we grew them together, they grew better than when they grew alone, which suggests that this is alone, this is together, and we count the specific copy numbers of each one of them. So this suggests that these two species are engaging in a mutualistic relationship where they somehow benefit each other. Now, of course, we wanted to see if it happens also in nature, and when we did that, we looked at the correlation of each one of these species to the presence of the plasmid, and the plasmid was highly correlated to the presence of both species. When the plasmid is low, both species are low, so the three are correlated in multiple different ecosystems, suggesting that these two species are positively interacting in a mutualistic manner. Then we went forward in an untargeted metabolomic approach where we fed each one of them with the spent media of the other to see what they eat and what they produce. They grow better on the spent media, and then we identified specific metabolite that one produces and the other one consumes, and vice versa, just by eating from the media. We ordered these metabolites from the shop, and we fed these microbes only by this on very, very minimal media, and these metabolites that they actually produce and consume contribute to growth, essentially suggesting that we look at a new paradigm on the way we were thinking about plasmids and stabilizing ecosystems. Maybe this is part of their role and the way to maintain them. And what we see over here is that two parties, they are in a fight. these two parties. One wants to kill the other, but the other has a defense system that prevents the toxin to actually kill it. And this interaction actually fosters an economical, profitable interaction between these two species where they cross-feed each other and contribute to the fitness of each other. So essentially what we're looking at is not just contributing to one host. It stabilizes a relationship which is mutualistic, that without the plasmid, it's actually very one-way ticket interaction. So in this case, it's kind of a higher order interaction that is mediated by the plasmid. So with this, let's summarize. I see that I thought that I will talk only 30 minutes. It's amazing. So we started by the effect of horizontal gene transfer on the hollow genome and the holobiome. We went forward by understanding who drives this or what drives it and what are the forces that are governing it. We learned that it's mostly neutrality. Sometimes we have selection that distills the most important genes, and then we drill down to a specific components that we understood that can stabilize a mutualistic relationship between two microbes, that this paradigm was not existent up until now. With this, I will first start by saying thank you to these amazing people that I'm just presenting their works. This was mainly driven by Sarah and Alva, which is not here. You saw her picture before. My friends and collaborators throughout this specific work: Ed Bayer discovered the cellulosome. Bill Martin, you saw, did the analysis of the verticality. Ron helped us with the plasmid. Jonathan Friedman with the neutral model. Eyal Gur, the biochemistry. Natalie, the microscopy. And Jens. And with this, I will say thank you.